Hello students, in previous class we studied about the tools of recombinant DNA technology. So today we are going to discuss a new topic, gel electrophoresis. Gel electrophoresis is a technique used to separate DNA fragments according to their size. DNA samples are loaded into wells at one end of a gel. So when we are loading the DNA sample, so that process we will call that as an indentation. And an electric current is applied to pull them through the gel. DNA fragments are negatively charged particles. So obviously the negatively charged particles will move towards the positive electrode. So this will be the principle followed for gel electrophoresis. So they move towards the positive electrodes. How do we find the existence of charge on colloidal particles? So the answer is electrophoresis. When an electric field is applied across two electrodes that are totally submerged in a colloidal solution, the particle tend to move towards one or the other electrode. This movement of particle under the effect of the electric field is known as electrophoresis. So what is electrophoresis? The movement of particles under the effect of the electric field. The particles which move towards the positive charge move towards the negative charge move towards the positive. So this will be the movement where the particle transfer, we will call the term as electrophoresis. Ferdinand Friedrich Rios was the first man to observe the phenomenon in 1807. The positively charged particles, that is cation, that moved towards the cathode were termed as catophoresis, whereas the negatively charged particle anion that moved towards the anode were termed as anophoresis. So electrophoresis, they are two. Okay, based on the connectivity, they are based on the connective charges. They are of two types. One is the catophoresis and other one is anophoresis. Whereas the catophoresis refers to the positively charged particle moves towards the cathode. Whereas the anophoresis, the particle moved towards the anode, is called anophoresis. This technique is carried out in laboratories to carry out DNA and RNA analysis. This technique uses a negatively charged to move protein towards the positive charge. So this electrophoresis is one of the type of electrophoresis is a gel electrophoresis, where the technique is implemented to carry out the laboratories to carry out the DNA and RNA analysis. Gel electrophoresis is a technique commonly used in laboratory to separate charged molecules like DNA, RNA and protein according to their size. Charged molecules move through a gel when an electric current is passed across it. The movement of charged molecules is called migration. Okay, the movement of charged molecules from one place to another place is called migration. Molecules migrate towards the opposite charge. A molecule with a negative charge will therefore be pulled towards the positive end, that is the opposite attracts. The gel consists of a permeable matrix, a bit like a sieve, through the which molecules can travel when an electric current is passed across it. The gel consists of a permeable matrix, a bit like a sieve, through which molecules can travel when an electric current is passed across it. Smaller molecules migrate through the gel more quickly and therefore travel further than the larger fragments that migrate more slowly. Okay, when it is a smaller molecules that migrate towards the end. Okay, so there will be the largest particle as well as the smallest particles of the DNA. Travel a shorter distance. As a result, the molecules are separated by size. What are all the steps which is involved in it? In this manner, DNA fragments as a solution are separated on the basis of size. There are several basic steps to performing gel electrophoresis that will be described below. Pouring the gel, preparing your sample, loading the gel, running the gel and staining the gel. So these are the steps which is involved in the Gel electrophoresis process. Gel electrophoresis. The DNA fragments are separated by forcing them to move towards anode under an electric field through a medium. So we were using the gel called agarose gel is used as a medium. Ethidium bromide is used as a stain for DNA, okay, where it shows a blue color. Then on exposure to UV light appear as orange color bands. Separated bands of DNA are cut off from agarose gel. So this is called elution. So what is elution? The separated bands of DNA where we cut from the agarose gel is called elution. These DNA fragments are used in recombinant DNA by joining them with cloning vectors. So the cutting of DNA at specific location. The purified DNA is cut by the use of restriction enzyme that act as a molecular scissor. Agarose gel electrophoresis is used to check the progression of restriction enzyme digestion. Next procedure is amplification of gene of interest using PCR, polymerase chain reaction. Amplification is the process of making multiple choices, multiple copies of desired DNA segment in vitro. Polymerase chain reaction involves 3 steps. They are denaturation, annealing, extension of primers. First we proceed with denaturation. The target DNA is heated to high temperature, that is 94 degrees Celsius, resulting in the separation of 2 strands of DNA, and each strands act as a template. Annealing. 2 oligonucleotide primers anneal to each of the single-stranded DNA template. Extension of primer. DNA polymerase, that is a tag polymerase, extends the primer using this nucleotide provides in the reaction. So tag polymerase is a heat stable, that is a thermostable DNA polymerase which is isolated from thermophilic bacterium named Thermus aquaticus. So next procedure in technology is ligation. The cut out gene of interest from the source of DNA and cut vector with appropriate space are mixed and ligase enzyme is added. So this result rDNA, that is a recombinant DNA technology, the process gets over. So next is transfer of recombinant DNA into the host. So once the rDNA is done, the ligated DNA is introduced into the recipient cell, makes itself competent to receive and take up DNA present in the surrounding. And last will be the result, obtaining the foreign gene product. So whatever the gene we were interested, the gene of interest we were inserted, we were going to gain out from that. The cell containing the foreign gene is cultured on suitable medium and the product can be extracted from the medium. Bioreactors are used for processing large volume of culture for obtaining product of interest in sufficient quantities. Bioreactor is a large vessel in which raw material is biologically converted into specific product under optimal condition. So this will be the final and the result where we get the multiple copies of the gene of interest where we created in the rDNA technology. Why is a buffer used in a gel electrophoresis? So there is a frequently asked question. Buffer in gel electrophoresis are used to provide ions that carry a current and to maintain the pH at a relatively constant value. So please make a note, students, okay? Thank you.